Hey everyone, today's set of notes we're going to begin unpacking the third line of defense and specifically looking at the role of T cells in the third line of defense. In terms of what your study design is asking you to be aware of, we need to make sure that we understand how helper T cells and cytotoxic T cells are used in the third line of defense to help combat against invading pathogens. So let's take a closer look at that now.
Let's do a quick recap of what we've done so far. So we have focused quite in depth on our innate or our non-specific immune response. We looked at our first line of defense, which was our chemical and physical barriers, things like intact skin, stomach acid, lysozymes in our tears, okay? All of which were designed to prevent the pathogen from ever entering our bodies. Now, if a pathogen was able to breach our first line of defense, that's when it moves on to the second line of defense. So our second line of defense is still part of our innate or non-specific immunity because it is treating all pathogens the same way. We've focused on the different cell types. So we've had our phagocytic cells, our natural killer cells, our mast cells, that sort of thing. We've looked at complement proteins and interferon, and we also looked in a bit more detail at the inflammatory response, which is our body's main internal mechanism for dealing with an invasion of a pathogen. So if your first and second line of defense fail, or they're not able to get a hold and I guess overpower that pathogen that has infected you, that's when your third line of defense comes into play. And your third line of defense is referred to as your adaptive or your specific immune response. The reason why will become a little bit clearer later, but in general, it is because this particular immune response will target the pathogen that has infected you specifically. So it will only target a pathogen with that specific shaped non-self antigen that your body has now associated with that specific pathogen. So for example, we've spoken previously about different pathogens or different cells really belonging to any organisms are going to have different antigens on their surface. For example, we might have a bacteria, and this particular bacteria, let's say it's E. coli, might have, I don't know, triangular shaped antigens on its surface. Our body will recognize those triangular shaped antigens from the bacteria as non-self. Some things from our second line of defense might try and destroy the bacteria that's within our body, but maybe that bacteria is replicating too fast and it hasn't been able to stop it from causing damage. Usually about 5 to 7 days later, that's when your third line of defense kicks in, and we'll talk about what's in your third line of defense in a moment. But essentially your third line of defense is made up of very specific targeted immune cells that will only scour your body looking for pathogens or anything really that have this specific exact triangular shaped antigen. If it doesn't have it, then your body doesn't care about it. Or sorry, those specific things of the third line of defense don't care about it. They will only be looking for anything that has that specific triangular shaped antigen, because that specific antigen has triggered your third line of defense to come into play. Most of what's on the screen here I've already kind of brought up previously with you guys. So obviously our adaptive or specific immune response is something that we develop throughout the life of an individual, and we'll talk about why it's developed throughout the life as we move through this. And it will only develop as you are exposed to a pathogen. So if you live a sheltered life and you're not exposed to different pathogens, your adaptive immune response is not going to be as good or as well developed as someone who has been exposed to different pathogens throughout their life. As I was saying previously, you need to have your innate or non-specific immune response, so your first and second line of defense, to have dealt with this pathogen previously before your adaptive immune response or your third line of defense is activated. As I was saying previously, it's unique to each pathogen or non-self antigen. So if you are infected with more than one different pathogen or more than one different non-self antigen, you actually need this response to be triggered by each unique non-self antigen. They're not going to, the cells that are activated in your third line of defense for one antigen will not be able to have the same effect on a different antigen. And the cells that we're going to look at today are the T cells, which are part of something called the cell-mediated response. We'll cover B cells and the humoral response in the next recording. Bit of a diagram here to point out to you how everything is connected. So we have our adaptive immunity or our third line of defense, and it can be broken into two pathways. So we have our humoral response going to the left and our cell-mediated response going to the right. We are focusing on the cell-mediated response today, which is focusing on cells that are referred to as T cells. Mainly three types that we'll be covering. So something called a helper T cell, a cytotoxic T cell, and then finally a memory T cell. We will cover humoral response in the next set of notes, so I'm not going to speak about them today. Our cell-mediated immunity, which involves our T cells, this particular branch of your adaptive immune system is activated when we are trying to defend ourselves against a pathogen that is intracellular. Okay, so this means that the pathogen that has infected our body hasn't only infected our body and our tissue, it's actually penetrating the plasma membrane and infecting our cells. So obviously our perfect example of an intracellular pathogen would be a virus. Okay, so every time you get the flu or a cold, or God forbid you get coronavirus, your cell-mediated immune response will be triggered, and this is the specific immune response that your body will use to try and fight off that virus that has infected your cells. Just a bit of background knowledge to link it to back to, I guess, the overall structure of your body. Your T cells mature in the thymus and migrate to your lymph nodes. If you're not sure where your thymus is, it's here in this diagram, so it's right in the middle of your chest. So your T cells will mature there and they will move to your lymph nodes. Your lymph nodes are I guess located at the parts of your body where your tissue is really, really soft. So up here on the inside of your throat, really, really soft. If you feel underneath your arms and into your armpits, okay, we've got lots of lymph nodes there. And on the inside of your leg or near your groin, you're also going to find that soft tissue, and below that soft tissue is where you're going to find a whole bunch of lymph nodes located. So that's where your T cells are stored until they're needed, obviously. In terms of what a T cell looks like, it looks like most cells that we have encountered before, but they do have a very unique feature that we haven't looked at before. So on the page here, you're going to see 3 different T cells. And each one of them, yeah, they might look like they are a little bit different because they're a different color or a different shape. That's just because they've been taken from different websites. The part of those images I want you to focus on are the receptors on the surface of the cell. Okay, so for T cells, they are covered in receptors on the surface of their cells, just like a regular cell. However, the receptors that are on a T cell are all the same. Okay, so if we're looking at this purple T cell over here, you will notice that every single one of these receptors around it has the exact same shape. Now, on a typical cell that is not a T cell, you would not expect them to have only one type of receptor on the surface of their cell. You would expect them to have hundreds, if not thousands, of different types so that they can interact with lots of different molecules and lots of different signals that are sent to them. Our T cells, on the other hand, have receptors that are identical because they are specific for one type of antigen only. So this T cell here that I've circled, this T cell will only be able to activate and perform its role in the body in the third line of defense if the non-self antigen that has triggered the third line of defense has a complementary shape to this receptor. If it doesn't, then this T cell will not be activated. If we look at this T cell here in the middle, we can see that it has the same shaped receptor across its entire surface as well. So again, its specific receptors are for one type of non-self antigen only. If that antigen does not have a complementary shape to these receptors, it will not be selected to be used or activated in the third line of defense. So the only way it's going to be activated to participate or to do its job in that third line of defense is if the non-self antigen has the complementary shape to the receptor on the surface of the cell. So for example, this last cell on the screen that I haven't crossed out, it's got a semicircular shaped receptor. So I might expect a non-self antigen to be circular in shape. And you know what? That kind of looks like it would fit in that receptor and therefore it would be complementary in shape. So this T cell, because its receptors are complementary to the antigen, it could be selected or would be selected to be activated and then carry out its role within the system. So now we're going to have a look at what exactly is its role in the third line of defense. We're going to start off with something called a T helper cell. Okay, so a helper T cell or a T helper cell, or sometimes you can see them written as the TH. They are all referring to the same thing. So T helper cells, I like to imagine them as maybe the general or the person that's in charge of your body's defense system or the army that is fighting against this pathogen. So they are these are the cells that are giving the orders, but very specific orders. Okay. T helper cells will only be activated, obviously, once our first and second line of defenses have failed. And the way that they can tell that these first and second line of defenses are failing or are not quite keeping up with the demand is by interacting with those antigen presenting cells that we spoke about when we looked at the cells of the second line of defense. So to remind everyone, the two examples of antigen presenting cells that we spoke about in the second line of defense were the macrophages and the dendritic cells. So their job was to encounter your pathogen that had non-self antigens. It would engulf it using phagocytosis or endocytosis and digesting it using the enzymes inside it. But the unique feature that they had was they were able to hold on to a small piece of that non-self antigen from the pathogen and display it on their MHC class 2 markers. So if we just look to the right of the screen here, this bit that I'm now circling is showing you an antigen presenting cell that has a small section of a non-self antigen displayed on its MHC class 2 marker. So this antigen presenting cell might have encountered this pathogen with the non-self antigens anywhere in your body. Once it has digested it and has a piece of its non-self antigen on its MHC class 2 marker, it will scour your body trying to find the corresponding helper T cell that has a complementary receptor to the non-self antigen it is holding. So this helper T cell here that is interacting with our antigen presenting cell, if it interacts with the antigen presenting cell and it does have a complementary receptor, we would now term this as the helper T cell has been selected. It has undergone clonal selection. Okay, it has been selected because it has the corresponding or the complementary receptor to this non-self antigen, and therefore it is going to be able to perform the third line of defense for this specific non-self antigen in an effective manner. Once it has been selected, it has undergone clonal selection, so it has a matching or a complementary receptor to the non-self antigen that the antigen presenting cell was carrying. That T helper cell will then be triggered to undergo something called clonal expansion. Now this is where the T helper cell will be activated and will naturally replicate itself really, really fast. So instead of just having one of these T helper cells that has a complementary receptor to the non-self antigen, you're going to end up with thousands of these in your body. As soon as these cells have been replicated, these newly created T helper cells are able to secrete cytokines. Those cytokines, their role, actually quite a few roles that it can perform, but the main ones are to continue to stimulate phagocytic cells, so the ones in the second line of defense, and sort of ramp up their production and encourage them to keep doing their job. They stimulate something called a cytotoxic T cell, which we'll talk about in a moment, and they also stimulate B cells. B cells and antibodies we'll talk about in the next set of notes. So just to quickly sum that up again, your T helper cells, they're the generals of your army. They're the ones that give out the instructions. Their instructions are these cytokines that they secrete at the end of this process. To get to the point where they're secreting the cytokines or giving out the instructions or the orders, they need to interact with an antigen presenting cell that has the corresponding non-self antigen on its MHC class II marker. If it is lucky enough to interact with an antigen presenting cell with that matching non-self antigen, we term that process as clonal selection. As soon as clonal selection has occurred, your T helper cell is then activated and starts replicating, which is known as clonal expansion. And once it has started to expand or replicate itself many, many times, those new replicated forms of the T helper cells are able to secrete those cytokines and give out the signal or give out the orders to the other cells and what they need to be doing.